And dear students of 12th class, and today in your botany we are going to take up the chromosomal theory of inheritance. As previously we were talking about the Mendel's laws, and now we are going to move a bit ahead, that is the post-Mendelian work on this genetics and hereditary traits. Whatever the progress was made after the Mendel's theories of inheritance, and the progress was made from 1900 onwards, when Mendel's work was rediscovered. And firstly, we will try to understand why Mendel's work was not recognized at that time, when Mendel gave put almost a new concept before the then known these biologists and the scientists of the world, that is the science of genetics. And Mendel is truly known as the father of genetics, as you already know, but due to ill fate of his work was not recognized. And now Mendel published his work on inheritance of characters in 1865, but for several reasons his work remained unrecognized till 1900. First reason was that the communication was not easy as it is now in those days, and his work could not be widely publicized. This was the one reason. Secondly, his concept of genes. Now we know the Mendel's factors as genes, or the factors in Mendel's words as stable and discrete units that control the expression of the traits and of the pair of alleles which do not blend with each other. It was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. He was challenged by then the scientists for the reason that there occur variations which are seen continuously occurring in the nature, and Mendel's theories could not explain that. And thirdly, Mendel's approach of using mathematics to explain biological phenomena, it was totally new and unacceptable to many of the biologists of his time. And finally, though Mendel's work suggested that the genes were discrete units, he could not provide any physical proof because of the lack of the technological support at Mendel's time for the existence of the factors, or say what they were made of. Still, Mendel was a genius and a genius of the geniuses who put forward the true picture of what lies behind the inheritance and transmission of the characters. And in 1900, three scientists, it was De Vries, Courant, and von Schirmack, independently they rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancements in the microscopy that were taking place, scientists were able to carefully observe cell division. And this led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division, as was proposed by Mendel about the genes. And these were called chromosomes, that is, the colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of the chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. If you recall that you have studied the behavior of chromosomes during mitosis, that is the equational division, wherein the number of chromosomes remains stable, and during meiosis, that is the reduction division, wherein you must be knowing that the number of chromosomes is reduced to half as meiosis occurs during the gamete formation. The important things to remember are the chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous sites on homologous chromosomes, and this is the pictorial representation of this meiosis during the germ cell formation in a cell with 4 chromosomes, and this can be visualized from G1, G2 phase, then meiosis 1, meiosis 2, and then the 4 germ cells are formed. You can see that here in the G1, at the G1 stage, it has 4 chromosomes. At G2, they become bivalent and are ready to get segregated, divided, and in the meiosis first anaphase, they are ready to get separated, and in the meiosis second, they get departed apart, and when on the formation of the germ cells, only one set of the chromosomes is transferred to each cell. The 2 chromosome pairs in the meiosis 1 can also are aligned at the metaphase plate independently of each other. That is, it shows the segregation of the chromosomes. To understand this, the chromosomes of the 4 different colors that you have visualized were shown so that you can understand that the gametes get only the one set of the chromosomes, that is half and half from either parent. And this is the chromosomal theory of independence, and you can see here the independent assortment of the chromosomes. As Sutton and Boveri argued that the pairing and separation of a pair of the chromosomes would lead to the segregation of the pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Later on, many more experiments were carried out by many more scientists, and one of them, who is the most famous, that is Thomas Hunt Morgan, who worked with the Drosophila melanogaster. There is the Drosophila melanogaster, and studied the character transmission in this fruit fly, what is called a Drosophila. And thanks. Have a nice day.